Jai Hind, welcome to BioAffairs. So today I will be teaching you about how to count the CFU per ml or CFU per gram or PFU per ml or per gram. So one by one I will clear your concept, but first know what is CFU. CFU, the name suggests, is colony forming unit. So colony, that means what colony? It is a bacterial colony made on the hard agar plate or nutrient agar plate or any type of media. Now, to understand the colony forming unit, you need to first understand the dilution factor or making dilutions, simple dilutions. So for your ease, I have drawn a picture. Please forgive me for my drawing skills. It's very bad. So let's continue. So this is a bacterial stock solution. It can be any bacteria. It may be E. coli, it may be Salmonella, Shigella, whatever you have. Okay. Now this is the original stock solution you have in a broth, nutrient broth, or in a saline condition. So it should be a liquid medium. Okay, liquid medium. Now from here it can be anything. It can be a 10 ml, it can be 100 ml, that doesn't matter, or a 1000 ml solution. The amount of solution here with the bacteria doesn't matter. But when you are making dilutions, further tubes, the factor of volume of broth or the media you have taken is very important. So suppose you have taken 1, 2, 3, 4, 5 tubes. Okay, 5 tubes. Now in those tubes, 9 ml of NB, nutrient broth, is there in each of the 5 tubes. Now what we will do now, from the bacterial stock solution, 1 ml will be transferred to the first tube. So this is tube number 1, or you can name them as tube A, tube B, tube C, tube D, tube E. So from the stock solution, 1 ml will be taken and given into the 9 ml nutrient broth solution. So 1 ml of the what? Bacterial culture. But here the 9 ml nutrient broth is sterile. It do not contain any kind of bacteria. So it is a sterile nutrient broth. Into that 1 ml of bacterial culture is added. So total volume become what? 10 ml. Right? So the dilution factor is now 10 to the power minus 1. Now you will say why? Why it is 10 to the minus 1? So 1 ml is transferred to make it a 10 ml. 1 ml is transferred to make it a 10 ml solution. So that is why 1 by 10 makes it 10 to the power minus 1 dilution factor. From there, from this tube A, now you are transferring the 1 ml to second tube, that is tube B. So now the dilution factor will be 10 to the power minus 2. Okay. So how it is going? 1 by 10, 10 to the power minus 1 into 1 by 10. So it is now 1 by 100. So it becomes 10 to the power minus 2. Okay. So from there again 1 ml to tube C, and from tube C to 1 ml to tube D to tube E. Likewise, you have to make a dilution of what? Bacterial culture. So every time you dilute, the dilution I am saying means you are deconcentrating the initial concentration or the initial amount of bacteria present in the stock solution. Suppose, for say I am telling you, this contains 1 lakh bacteria, that stock solution. So here it become 50,000, it becomes 30 here, 20 here, and 1000 here, 500 here. Just to say, just consider the example. So this is the purpose of dilution, is to dilute the number of bacteria present from the stock solution to the last tube. Okay. So likewise 10 to the power minus 3, 10 to the power minus 4, 10 to the power minus 5. So I have taken only 5 tubes here. You can extend this to 10 to the power minus 10, 10 to the power minus 11. That depends on your experiment. Okay, but I have taken it to 10 to the power minus 5 only to make it simple for you and make you understandable. Now if you plate them, so we need to count the colony, but how will you count? You have to plate the bacteria into the nutrient agar plate. Okay, now how will you take that? Now from there, suppose you have taken 1 ml of bacteria, 1 ml of solution from 10 to the power minus 1 dilution factor, DF dilution factor, into a nutrient agar plate. Now during this process you can use either spread plate or pour plate. That depends on your experiment. It has nothing to do with your process. Okay. So when you plate it, the 10 to the minus 1 dilution in nutrient agar plate, and keep it for overnight, 16 to 18 hours of incubation, and next day you will observe the colonies. Okay. Likewise, you will take 5 plates, 5 nutrient agar plates. 5 nutrient agar plates will contain samples. How much of sample? 1 ml of sample from each tube. Okay. So plate A, plate B, like tube A, tube B, from tube A to plate A, tube B to plate B. From there you have cultured. Okay. Next day you will count the colonies. So suppose this is the colony count. So you will observe the highest dilution. What is the highest dilution? 10 to the power minus 5 contains the lowest number of colony. And the lowest dilution, that is 10 to the power minus 1, will contain the highest number of bacteria. Most probably you will not observe any colony in this plate. You will observe only a confluent growth because the number of bacteria is too much to count. Now considering this fact, our eyes cannot calculate or cannot count more than 300 of colonies. More than 300 of colonies cannot be counted. Okay. So for the ease of counting, we will take the lowest number of colonies present in the last dilution, serial dilution. So I have written arbitrarily and considering the amount. Suppose in the dilution factor C, or the plate C contains 350 colonies, plate D contains 200 colonies, and plate E contains 120 colonies. Say for example, I am telling you here, not the exact part I am telling you. So exact experiment is different. You may get any number of bacteria. Now the formula you have to apply to count the CFU per ml is number of colonies counted, number of colonies in the plate. Here it is 120 colony. Sample volume, what is the sample volume? You have taken 1 ml from the each tube to each plate. So the sample volume is here 120 colony by 1 ml of sample, that is the sample volume from each plate, and dilution factor, it is, we are taking 10 to the power 5. What plate we are counting? That is plate E. Plate E comes from 10 to the power minus 5 dilution. So the dilution factor is 10 to the power minus 5. Right? So as per the mathematical formula, if we break it 120 into 10 to the power 5, again 112 into 10 to the power 6. Now 1.2 into 10 to the power 7 CFU per ml is the answer. Very easy to calculate. You can change this value accordingly and you can calculate. No problem. Okay. So you can take here 200 colonies. The calculation will be changed accordingly. So it is a simple mathematical formula. Okay. Now considering this fact that we will take the highest dilution with lowest colony, it will give you an ease of calculation. As you can see in this plate, a ease of calculation, very less few colonies, not too few to count, but a medium plate count like 50 to 100 or 150 is a good count of colony to take in this or to put in this formula. So CFU per ml. Now 1.2 into the power 7 is the CFU per ml. Now one thing I want to tell you, that if you change this volume pattern, means, let me show you, from the initial stock, considering this 1 ml, you have what we have taken 1 ml into 9 ml of broth solution. So if you take 0.1 ml of stock to 0.9 ml of broth, so total volume will be 1 ml or 10 to the 0 minus 1 dilution factor. So whatever you take, dilution factors will be equivalent. 10 to the 0 minus 1, 2, 3, 4, 5, 6 like that. So here 0.1, 0.9. But so only one difference, if you calculate this amount in this formula, here the sample volume only will be changed. So I have taken 1 ml because initially we have taken 1 ml sample from each tube. Now if you take 0.1 ml of sample, here the 1 ml will become 0.1 ml and the calculation will change accordingly. This is very simple. Nothing to worry about. Okay. So I hope you understand the CFU per ml. Now let's understand the CFU per gram. Simple change and everything will be okay. Suppose in I'll wipe off these things. Now instead of bacteria, you have diluted 1 gram of soil. Suppose soil you are counting. But instead of bacteria, 1 gram of soil is dissolved in, suppose 50 ml of water. That is the stock solution. It is the stock. Right? So 1 gram is dissolved here. Now from there, we will again do the dilution. From 1 gram of soil you are counting. Everything will be same. Here, you will do CFU per gram. So, if you count it from the colonies, because 1 gram is transferred and diluted one by one, again everything will be same, but the ultimate result you can transfer it to CFU per gram. Because the initial sample you have taken 1 gram of soil. From there you have counted the number of bacteria. So you can easily write that is CFU per gram. So CFU per ml, CFU per gram. Very easy. Now I will come to plaque forming assay or plaque forming PFU per ml. Like bacteria, which you can observe colonies on a nutrient agar plate, in case of virus, when I am telling you virus, actually I am telling you about bacteriophage. Because in a normal lab, in a normal environment, we can culture the bacteriophage because bacteriophage attack what? Bacteria. So easily you can culture them in a lab condition and see the plaques. So what are plaques? As you can see, plaques are nothing but the halogens around the bacterial lawn. So the zones will be counted as each plaque, one plaque. One plaque considering one virion. Okay, one virion family from one virion. So one plaque, like the CFU, PFU is there. So plaques are looking different, as you can see here. Plaques will be looking different than colonies. And plaque represents virus. It can be, if you consider, I am telling you about plate, because bacterial lawn is there, bacteriophage will attack the bacteria and will show you the plaques. If you consider the animal viruses, then you have to take the animal cells, because animal viruses will not infect bacterial cells. It needs animal cells. which is very difficult to see in proper or in BSL2 lab or a BSL2 lab. Not the problem is BSL2 lab, the problem is the type of culture system. The animal cell culture is totally different than bacterial cell culture. Okay, so the ease of counting, I have taken this bacterial example, but the total formula is same for animal viruses as well as bacterial viruses. So let's come to the PFU per ml or PFU per gram. Now consider the dilution factor again. You have to dilute it again, but here we will dilute the virus, not the bacteria. So you have a stock solution of bacteriophage or the bacteriophage, any kind of bacteriophage, you can say the E. coli phage, maybe the salmonella phage, any phage, and the host bacteria that depends upon the viral nature. So from the stock solution, again you are taking 1 ml. So forget it. In the last CFU, I have told you about 1 ml. Now I will show you with 0.1 ml. Okay. So let's consider 0.1, 0.1, 0.1, 0.1, 0.1, and this is 0.9, 0.9, or 0.9, 0.9, 0.9, 0.9, and 0.9. That is 0.1. Okay, so 0.1 ml of the viral stock solution has been transferred to the first tube A. Now this tube contains 0.9 ml of buffer, PBS buffer. Say phosphate buffer saline I have taken, or normal saline you can take, no problem. Normal saline, okay. That is body saline, normal saline. So in the normal saline it is there 0.9 ml, and from the stock solution 0.1 ml of the virus has been transferred to the tube A. So again it is 10 to the power minus 1 dilution. So likewise 0.1, 0.1, 0.1, and ultimately 10 to the power 5. Again I have taken 10 to the power minus 5 is the highest dilution factor I am taking here, and the phage become diluted, or the virus become diluted with the help of a saline. Now from there you have to plate the virus on a bacterial plate. Now how will you do that? First you have to take a nutrient agar plate, right? Then you have to take a bacterial culture, that is separately you have to have in your hand, and you have to make a soft agar. Okay, in that soft agar you will just pour 0.1 ml of the phage plus 0.1 ml of the bacteria. So I am telling you again, a nutrient agar plate, sorry for the color, nutrient agar plate plus soft agar, that is 0.8% soft agar. I hope you know that soft agar, it is a semi-solid condition of a nutrient agar media. Now in this soft agar you will give 0.1 ml of the phage, which is taken from tube A, and 0.1 ml of bacteria that you have already. Which bacteria? The same host of the virus. So if you are taking E. coli bacteria, then the phage will be E. coli phage. So 0.1 ml of the E. coli phage will be given to this 0.8% soft agar. Now this soft agar will be poured on the nutrient agar plate, and immediately you have to circulate the plate so that a lawn will be prepared. Okay, you have to, before that you have to mix the soft agar and then you will pour it. So you will have a almost very equal concentration of bacteria and phage present there. Now you have to keep it for overnight, 16 to 18 hours of incubation. Likewise from plate 1 you have done. Now from plate B, from plate C, plate D, and tube E. From there you have prepared plate B, C, D, E. Everything is same, but you are adding one more factor, that is including the virus, you are adding the bacteria in the soft agar medium. And this soft agar is again poured upon a nutrient agar plate. So you will have a lawn of bacteria and bacteriophage above the layer of nutrient agar, hard nutrient agar media. So next you will observe this picture, a plaque embedded plate, but not all the dilutions or not all the plate from A to E will show you, because there is a factor called MOI, multiplicity of infection. I have made a separate class on this multiplicity of infection. So if you have not seen it, please go to the information section, I button, I will give it the link, and also in the description you will get the playlist. Now, due to high MOI, multiplicity of infection, in short I am telling you one line, that the number of virus you are applying for the number of bacteria, right? Like for one bacteria you are applying one phage or one virus, or you are applying 10 virus or 10 phage. That is called MOI, multiplicity of infection. This is a very important factor when you are considering the plaque assay or PFU per ml. So likewise in the CFU, if you remember, the 10 to the power minus 1, 2 will not give you a single colony, it will give you a confluent growth. Here in case of PFU, you will get a clear nutrient agar plate. So next day you will observe, you will see a hazy kind of layering appearances in the plate, but you will not see any bacteria or the plaque, because the number of phage will be higher compared to applied to each bacteria present there. So it will totally lyse the host cell and will not get any kind of plaque or a confluent growth. But this scenario will change when you increase the dilution. So considering this, again I have taken 10 to the power minus 3 will give you 300 plaques, 10 to the power minus 4 will give you 200 plaques, and this will give 120. So now this time number of plaque is 120, right? So I am taking dilution factor 10 to the power 5, that is the dilution factor 10 to the power minus 5, and the sample volume is 0.1. Here we are applying 0.1 ml of phage. Okay, we will not consider this bacterial volume, we will consider the 0.1 ml phage. So the mathematics, simple mathematics, it is 10 to the power minus 1 into 10 to the power minus 5. So in the next step, 120 into 10 to the power minus 10 to the power 6. If you change the equations, now 1.2 into 10 to the power 8 PFU per ml is the answer. So I skipped one step here. So this is the number of phage present in this tube. So in dilution factor 10 to the power minus 5 tube, the number of phage or plaque forming units present is 1.2 into 10 to the power 8. It is as simple as water. Same thing, same. You have to just remember this critical factor during lawn preparation, and everything apart from that is same. Okay. So again, PFU per gram, I have told you. So if you want to calculate a PFU per gram in a tissue, suppose we have taken an animal model, there you have applied the phage and the host also. Now you want to see whether the phage is there in that tissue, suppose intestinal tissue. So again, 1 gram of the tissue will be diluted or will be solubilized in a perfect buffer or saline. That will be your stock, and from there you will take again dilutions, different kind of dilutions, whatever you want, 6, 7, 8, 9, and from there you will count with this formula. So PFU, I have cleared your CFU per ml, CFU per gram, PFU per ml, and PFU per gram. So in some portions I will give you pictures in the side by side, so it will clear your concept more. And if you have any doubt, please ask me in the comment section. You can mail me, you can join in my Telegram channel. So this kind of conceptual video takes a very much effort. So if you really understand my class, then please like and subscribe my channel for further enriched videos. And you can check out my playlist also. There are several videos which might be helpful for your studies. So thank you very much. Signing out.